to create awareness among our senior officers to initiate some of the conservation program, because in this the forest resource, forest genetic resources, there are two aspects. One is the adaptive potential of the genetic resources, genetic materials, that the nature takes care of it because that is part of the evolutionary process. But artificially human beings have to then they have to interfere and say they manage in such a way that we sustainably conserve as well as use these resources for the benefit of the mankind. So these two objectives are very critical for the forest managers to balance. When you were all joined the service, you were asked to do tree improvement program, how to increase the productivity of the timber species, how to increase the productivity of non-timber forest products. Then you were taught only about how to do clonal propagation. Actually, the Aracruz method of clonal propagation was initiated by me for the first time in India, and the Brazil technology. Similarly, we did a lot of clonal propagation. We were never asked to do anything classical tree improvement programs. Of course, Dr. Kedarnath had initiated in 1960s the tree improvement in Teak. He collected a lot of plus trees and also assessed the genetic variability for various the timber characters. And he did a lot of I think the tree improvement program by establishing seed orchards. They are called seed orchards. So the selected plus trees which were tested and assessed, they were planted in a given the statistical design. He employed the design called a simple lattice design of 11 by 11, where each species like combining with the neighbors of other the species which are in that particular replication, so that you ensure that the pollens from every plus tree is allowed to fall on the female tree of the genotype that he had selected. So this was a very classical tree breeding program. But for this, none of us were exposed to the molecular genetics manipulation of this tree improvement programs, or identifying or assessing or documenting. Nothing was done. So we are very lucky that we are reinventing the wheel. We knew what is forest, we knew what is resources. Genetics was never there in between. Now they introduced the genetic forest, genetic resources. It's a great thing to happen. So coming to what exactly forest genetic resource, that Government of India or policymakers means is, forest genetic resource is nothing but it is a hereditary material that enables the woody trees and also associated species to survive, maintain, and adapt to the new environment, and also provide the economic benefits to the human kind. So this is a very simple FAO definition. However, what it underpins is the potentials of genetic resources. I mean, we were not exposed actually. Now we have understood what is the potential of it. It is sitting there in the forest as a future, the use. We do not know what will be its, future use. For example, climate change is happening. We may have certain genotypes, within our ecosystem that may be useful, for example, to be adopted in a stressed environment. For example, the temperature may fall in Himalayas. Then how the lower elevation species can really go to the top, and also some of the land species which are very close to Himalayas can move to the lower elevation places in Himalayas. So these kind of species migration and movement that will happen only because of the adaptation. That adaptation is possible only because of the species has the capacity to change through mutation or through recombinant. We're all very familiar now. The whole world has become geneticist. Every man is now a geneticist because he knows how corona is changing, how corona mutating, how it is changing its RNA. People know what is RT-PCR, reverse transcriptase, polymerase chain reaction. Everyone knows. Earlier days, only people who knew molecular genetics knew what is PCR. They knew what is isozyme studies. But now everyone knows what is RT-PCR. It has become like an alphabet, RT-PCR, then PCR test, everything. So I'm very happy that now people are very familiar with these genetics terminologies. And now, with this background, we should try to adopt and try to strategize. How do we really manage these genetic resources not only from the point of conservation for the future needs, and also to meet the current needs. That is how sustainable management of forests are defined. Now let us come and see the second slide. Second slide. The evolutionary significance I already explained, because it is sitting on the potential. We need this evolutionary potential genes to be used by us, or to be some eminent geneticists for future use. So in this context, we try to classify the forest types. When I was in actually ICF for years, this chair of excellence, and see whether we can really identify the genetic variability that is existing among the forest genotypes. The champion and said, no, they attempted in 1965 to classify the forest type based on the morphological character, that is physiognomic character. What they did was they took one bioclimate parameters, that is tropic, subtropic, temperate and all. That is the first group. And the second character they selected was based on their appearance, whether they are evergreen, whether they are semi-evergreen, only in some part of the year they appear semi-evergreen, whether they are moist deciduous, they shed the leaves, but still they are moist in their sides, whether they are dry deciduous. So based on the rainfall and based on the latitude, as you move up from the this equator to the top, actually it's not even your rainfall, not even your other factors which shows what is the forest type. It is actually latitude is a single factor. You tell me the latitude of any place, I will tell you what is the species, because that is how the latitude and species this species the concentrations are correlated. Because as you move up, what species you find in subtropical region, you know what species you find in temperate regions, what you find in Arctic region. So it is a very simple, the correlation between the latitude and the this thing. But rainfall also, because because of latitude, rainfall also play an important role, because consequently, this your the latitude positions, your rainfall in the tropical region is very high. And as you move up subtropical, it may reduce. But however, as you go more and more towards an Arctic region, your humidity will increase. So they actually decide the species. So based on these character, our champion and set people, they try to classify. India has a very unique forest types of 250 types. Can you believe it? So it was actually 6 major groups, 16 groups, and also there are subtypes of nearly 240. What they did was, number one was the this your latitude, that is tropical or subtropical. Number two was whether how they appear, that is physiognomic character. Number three was they went to the species level. They went to the extent of telling very moist teak, dry teak, like that. There are 8 classification of teak within these 240 types. Similarly, sal also the same pattern they have copied. Very moist sal, then moist sal, dry sal, very dry sal, peninsular sal. Species is same, but depending upon whether he went in the morning, if you see the site is very moist, he has called it as moist. When he has gone in the afternoon, if it is very dry, he has called it as a dry sal. Like this, based on these characters, which are not really very scientific, there are a lot of overlappings are also there, but they have tried to classify based on certain characters, so that they have similar the taxonomical features and also similar ecological amplitude. These are the 2 criteria they have used. But based on that, we also took the champions classification of the Bible. We try to manage our forest based on that, and all our management interventions, all our management objectives were only based on the classification that were recommended by Champion and Seth. But however, in their preface, they had written Champion and Seth, you please, guys, you please go back and look. I did not have the time to, because he came in 67, he goes back in 38, goes to Oxford Forest Institute, where I also went. Then he was the personal assistant Slitch, who was also a Oxford Forest Institute head. Of course, he married his sister also. He stayed there, he comes back in 64. Then 2 years he stays, but only 3 months he tours entire India. Wherever train connections were there, he went. But luckily, when we went to revisit his forest types, as requested by Champion, we went to all the sites as he classified. We took the measurements, we measured the areas, we classified the species, we also estimated the biodiversity value of those. We compared those sites with what champion, whatever, wherever photos were there, we compared with that, and we found a lot of differences in that also. But he was very humble in telling in his preface, any forester of in India, if he goes and recheck, verify, and deep this this forest of India, I'll be very happy. Now we know what is happening in all over the world. All over the world, they do not use the taxonomic characters in the classification basis for the forest types classification. They go because satellite images are used to classify the forest. They go only with physiognomic characters. They go whether it is broad-leaved or coniferous. In the broad-leaved, they go for some of the characters like whether whether it is temperate broad-leaved, whether it is tropical broad-leaved, like that. So they have any you go to any international classification of the forest types, which are based on the satellite images, reflectance values are used, and any point of time you should be in a position to get the images of those forest vegetation, and also you should be in a position to assess which forest type it belongs. So when the whole world is moving in that direction, FAO and IUCN are wondering how are you going to incorporate and enter the forest types of India, which champions have prepared, and they are based on the tropical characters and based on the evergreenness, semi-evergreenness, then Euphorbiaceae forest, then cane breaks, then also bamboo breaks. So these kind of the fourth level of subclassification subtype. We have 5 types actually. If you really divide by entire 100 240 by 5, you come to only 60. Again 60, if you take away north and south, you also find Dipterocarpus turbinatus in Assam. You find Dipterocarpus indicus in Karnataka, or Dipterocarpus macrophyllus in Andaman. But you call northern tropical evergreen giant and you call the southern tropical wet evergreen. It doesn't make any sense at all. But still, it is a Bible. We have to follow it. We have not distorted it. We have retained it. We went. Now, Forest Survey of India is trying to identify whatever he has identified in his famous book, whether those forests are there. 36 forests are missing, subtypes. We went and found out Laricia is not found at all. Then there was Euphorbiaceae. I spent 2.5 days in actually Mizoram and other places. I did not find Euphorbiaceae forest. So Karnataka, for example, see around Dehradun, can you believe we have got 42 subtypes of forest? 42 for each site of different elevation or different the group of species. They have been classified as a forest. Your type means it should be the same characters. If you apply, it should repeat. For example, if they say tropical forest, tropical 11 degree, 73 minutes, then 11 degree, 15 minutes, it should appear in the same. That is how there is some method in the madness. But however, if you within 11 degree, 73 minutes, we have got 6 or 7 forest types of subtypes, which are actually for the species level. I wonder now how whether international bodies like IUCN or WWF is finding it difficult. They are given their own set of classification for the whole world. But India also falls into that biome geographic reason. So that is how we try to classify forest types, and we found wonderful the result of forest types of India. I was very lucky to visit most of these areas. There are a lot of changes. For example, 131 species were actually recorded during champion writing the book, maybe in 1960. But last when we found we have found 7 or 8 species more. The same species were found in different places, but they were again they are found in the say 107 or 8 species. Similarly, in Karnataka, we found the species which were not mentioned there. Actually, we found in that list of those forests. So like that, so we found a lot of variation. We do not know whether it was erroneous or that these species have later on appeared, because 1960 perhaps the forests were more degraded than it was. Thanks to Indian Forest Service, the very the committed cadre, the best cadre, forest cadre according to FAO, according to World Bank, in the forest management, the most the highly qualified people, highly technically qualified people. No country can really boast of such a high caliber people with PhDs, with post graduations, with postdoctorals, and with very high IQ. Thanks to the salary structure, people they join here and very brilliant people. So they have really contributed in really conservation and protection of these forests in all over the country. And we were really wonderstruck to see the changes that are happening. Some of the evergreen forests which were in degraded state and actually which were actually in the semi-evergreen and moist glacier state. That how the progression and retrogression of forest takes place. Champion has given a nice figure if you go to the 73rd or 74th page, where the evergreen will gradually degrade into semi-evergreen and semi-evergreen will degrade into moist deciduous like that into goes. But if you reverse, if you provide protection, the reverse process, retrogression takes place, and pioneers will appear, slowly then it gets into deciduous types, then moist deciduous type. So we found most of the protected areas, thanks to the fire, protection, thanks to grazing protection, thanks to, your, other, kind of, the, so stopping, this removal of, trees and encouraging the regeneration. We have found a lot of, the evergreen species reappearing in the semi-evergreen forest, and moist deciduous species are appearing in the semi-evergreen. So like that, there is a species migration in the last 20-25 years towards the evergreenness. So this is a very, very, wonderful finding that we have found. Even in Andhra and Telangana, I heard just now a speaker speaking on Telangana. We found, moist teak actually in some of the area. I presented this, in Tirupati when Mr. B.S.S. Reddy was PC of my batchmate. He was very happy, and we also showed, some of the photographs of that. And teak ecology also in Kerala is changing, not because rainfall is changing, because of the management intervention is changing. So the management interventions coupled with the climate change and also other the factors, protection factors, the forests are changing in India. The number of species are increasing, the trends are increasing. And Karnataka also we have found even the solar forests which were completely discarded, were not at all included in the management plan also. Now we have seen a beautiful species of this lower elevation species slowly migrating, and the solar forests also getting covered with the tree species. So these changes and soil we found northeast towards northeast, the soil is spreading. This also has been not only in our studies we found out, also there were some IIT, I think Kanpur or somewhere. Those people also they have made studies through satellite images. They have found they have compared with the earlier maps of soil map, and they shown ripples of soil, how the this soil is expanding. And we found in Orissa also the soil is spreading towards the coastal area. So this forest types of India revisited was a very fantastic experience, and we have documented 600 pages in a book has been brought out, and we have published it also. We also published in the your Oxford Journal of the forest, this common forestry, this thing. It has given, and a lot of people are also asking the reprints and other people. So it has given is not only we are trying to recommend what should be the real practical classification of the forest for the people to follow, and also we are trying to identify what was there or what is change matrix now, and how the species have changed, and how because of the management interventions. Also we are compared with the climate change factor, because we have found that in particularly Maharashtra or Mahabaleshwar, it was 7,200 was the 100-year rainfall. We have found now 5,200 rainfall. There is a gradual shift in the rainfall pattern there. So there, how the species are adapting to the low rainfall, how the high evergreenness is going off, that has been recorded. We have tried to compare with the rainfall changes across India and also the forest changes, how they are responding. So, I mean, for various bioclimate, for various management interventions, we are trying to identify and we are trying to give reasons what is happening, which species are spreading, which species are shrinking, and where we are actually gaining and where we are losing. This is a very wonderful exercise. I think we should keep doing it. So this is a very broad, level of, trying to identify the genetic variability that exists among the forest type. That is what FAO also defines. FAO defines forest genetics resources as it is the genetic variability that exists in the forest types that have value as resources to derive the benefit to the mankind. That is how FAO is given very simple. There are three components. I mean, it is a genetic, no doubt, hereditary material. So no doubt, it has a resource value, and also we should also it has a potential of adoption. These are the three main components. So accordingly, these forest types of India have been, re-looked into, revisited. I thought because I had done, little work in forest types of India, I should share as a part of forest genetic resources. I don't know how relevant it is. If there is additional information, if you have, if you did not know, for example, okay, see, what are the two main species? We have, 16,500 species in India. We have found out of 16,000, the importance value index is highly highest for the Asteraceae. No one knows, Orchidaceae is the largest number of, species in India. For example, rhododendron itself, has 80. Asteraceae has got nearly 800 species in India. Of course, all over the world, Asteraceae has got 30,000 species. So we have to link it to the evolutionary trend of, this genetic variation to the Eurasia and also our, the Gondwana period, where we have been finding a lot of, fossils, where we have found even eucalyptus also in Madhya Pradesh, because it was part of the Australia and, India. Naturally, when the Gondwana it got split in and joined, this Himalayan, this European region, then suddenly these species which were there 50 million years ago are found even today. So species, now we are in the realm of the Indo-Malayan region. After the Gondwana period, after Indian part joined, this Himalayan, this thing, now, the species, that were there, the Himalayan, the tropical forest, they have started moving. That's why, Dipterocarpus is another, very, because all our North India is completely Dipterocarpus here. Don't tell, only North India of Sol is occupying. Even come to, they were South India, where entire Western Ghat is Dipterocarpus, whether it is Hopia, whether Veteria, whether Dipterocarpus indicus, whether Andaman, they're all Dipterocarpus. Even the drier areas, we have got no Dipterocarpus, Soria, Talura. You go to Assam, you have got Soria Assamica. So what has happened is, these species of Indo-Malayan region, they have spread only after the, I think, glacial period of maybe 20,000 years. We have history of only 20,000 years. We do not have the history of, 10 million, 15 million years of some continents have. Because ours is a new area, it came to a new latitude. As a result, we have new species combination and new species distributions, which are, of course, intervened by the man. Indian forests are now heavily worked, not only during, the colonial period, before colonial period also, the Mughal period, people have worked, they have removed the huge timber, they've exported to European countries, they have tried to maximize, the single monotypic species, and they have removed, all the associate species. Same thing happened in South India, where the teak was the major, preferred species. So we have lost, a lot of biodiversity, lot of gene pool, thanks to the management. However, now some of the areas like, tropical evergreen forest, do not have, much of hardwood species. We have, still, the intact of, the species combination. If you put, the area and species cover of, tropical evergreen forest, you find nearly 150 in, 1 kilometer area. Whereas, if you put the same thing of, area and curve species in the moist deciduous, it comes down to 60, 70, 80. Why? Because not that we did not have, the rich species diversity in the moist deciduous or, in other areas. Because we have exploited, we have converted this forest of, high genetic diversity into narrow genetic diversity of, monospecies, either for the teak, either for the Hardwickia bainata, or either for, Soria robusta. So we have lost, we have 150, 200 years have been spent in, converting, our, the non-wood species, so non-hardwood species into hardwood species. And as a result, we have to pay the price. We have lost the genes. Gene erosion, I was told, someone was telling, gene erosion has taken place in the Hardy-Winbury equilibrium. Polymorphism happens because, if they're widely distributed. If, one area is lost because of fragmentation or because of its removal due to logging, you lose the gene. So these kind of, the imbalances, do, contribute for the loss of genes, which is not really good. Of course, the world does not wait, worry. It will not spend sleepless night if they lose, some of the, Hardy-Weinberg, equilibrium, some of the alleles, because when they are losing, the big animals, when they are losing, the big, when they are lost, dinosaurs was lost on its own. Now we are losing elephants, we are losing, the tigers in most of the countries. When you lose, bigger animals in a bigger, genetic, constitution, we are not worried about, losing, small, species, which is very small, which has no consequence according to their knowledge. So now to reinvent the wheel, to reset, we have to come back to the process of see this resource genetic foreign genetic resource conservation strategy. Conservation, everyone is very well aware of it. We have to have first assessment. Unless we make assessment of where are we have no forest types 1968. Later on, have we made any within that? We have not made any. Diplocarpus is, particularly over Indonesia, South America, they have made so much of genetic studies. They have gone to the molecular level of isogyme studies, and they have mapped genome mapping. They have done. They have done which species which is has very narrow ecological amplitude, which one has broad ecological amplitude. These kind of the things that they have. But in India, we have done classical tree breeding. We have not done any genome analysis. We have not done, except Vasudevan, I think, is the next speaker. He will be speaking. I know what kind of genome analysis we have done, DNA level and also PCR level. We did for some of the classical tree breeding species in Karnataka for teak. We tried to do whether there any polymorphism is there, and to see whether any similarities there between the clones. We try to do D-square analysis, genetic distance between the species, and also within the species we have tried to do. I think we published jointly a paper with Vasudev. He's one of the best foreign geneticists I have seen. I think we'll be sharing. Similarly, this A3 people did in Sandal. Sandal also, it was IFGTB should all the credit should go. They have done wonderful work. Classical tree breeding, they identify the clones, they did the cloning, they did the seed orchard, they did the specific combining ability, they did the genetic general combining ability. They have estimated SCA values, GCA values. They have also found out which is the most prolific open pollinator. This kind of information we have, and we have also seed orchards. So in Karnataka, what we did was we did not have the mandate. My mandate was those days clonal propagation. We did a lot of clonal propagation till 90. Then later on 90, we changed our strategy. We came back to this little bit of genetic tree improvement studies. Then we tried to identify to the assessment of some of the major hardwood species. We first identified the plus trees. That is the first step in any genetic tree improvement program. Then we started evaluating them. Then we started converting them into seedling seed orchard. Seedling seed orchard in the sense where you collect the seeds from the plus trees, and you try to weather seedlings. Whereas in case of clonal seed orchards, you do not use the seeds. You actually do the vegetative propagation. The whole plant is copied along with the 100% genes. In case of seed orchards, in case of seed, you are not copying. You are collecting the seed. So seed may not be behaving like your mother tree, unlike unlike its syzygium. Syzygium is a polyembryony. It behaves like its mother, but others seed will have only 50% probability. We went for seed orchards of teak, and we also went for I think 3-4 places we did. So we have done in the teak and the sandal. We were shocked to see some of the results of the polymorphism study. We have lost a lot of alleles there because of the sandal smuggling was very rampant in the border areas. Border areas had I think lot of very good sandal trees with the different the gene combinations. And we have found there is a loss of gene, erosion of genes. That was a very good study. I think I was also on the part of paper Ganeshaya, then Gauri Shankar. We did that studies. That was really shocking. Similarly, the way we harvest, if we remove all the seeds, if these seeds are actually they are contributing to the regeneration and also perpetuating the gene genotype, then we'll be losing if you take away all the seeds. That is happening the teak stands. That is happening in one of the many of the seeds which we collect for our regeneration purpose. So we have done seed improvement of nearly 1,000 hectares of teak. It supplies nearly 1,000 tons. We 1 ton per hectare like that we calculated. We have in around 1,000 hectares of teak seed orchard. We have Heritable marsupium. We identified, of course, we did only in terminalia some of the DNA studies, isogyme studies. One Rani Krishnan was there. She was very keen on doing this DNA isogyme studies. We did in terminalia to identify any differences are there. Then we also did took up some at the molecular level some studies in some of the endemic species like Garcinia. So these were actually endangered. We wanted to know what the mating system there, why the seeding is not happening. Particularly there was some tree species. I have forgotten those, but there we had a seeding problem. So the trees were there, But some of them, were dioecious, some were monoecious. There was some kind of a mating problem, mating system problem. So unless we have the knowledge of, whether they are open pollinated, whether any problem in the floral biology. So are the differences in the time of pollen and also the maturity of the gynecium. So these kind of synchronization of gynecium and andrecium. So these knowledges for each species is essential. Unless we cannot really manage forest ecosystem based on the species characters, based on their adoption, based on ecological requirement, and also their future evolutionary value. We cannot do. Therefore, all this knowledge is necessary. And I am sure the new management interventions and the new forest policy and also commitment this sustainable development goals by 1930. The Karnataka has done quite a bit of work in the last 15-20 years during my time, because luckily I was I was student of genetics. I knew what is genetics. I knew what is molecular genetics. So because of these knowledges, we tried to initiate classical breeding. We tried to develop seed orchards, seedling seed orchards. Then also we collected a lot of germplasm. So we have germplasm banks, a few germplasm banks which are located in different places, very close to nurseries, particularly dry zone species. And these the cyan materials are used for multiplying, and also so giving grafted materials. Particularly in tamarind, we have done a very extensive work. We have been doing for the last 20 years. Jackfruit we have done. We have done in Sitapal. We have done in, I think, Feronia. Then we have got Syzygium. We tried to do grafting, clonal propagation. We stopped it because we knew it's a poly embryonic, and where a nucellus cell will be becoming a 2n. So it will be carrying entire set of chromosomes. Therefore, we did not follow it. So at least around 15 to 20 species, we did standardize the clonal propagation, micropropagation, establish the germplasm banks, and also we are supplying this propagated materials which were really endemic. For example, Artocarpus lepucha we tried to multiply, because that was also listed. Garcinia we tried to. Then also in mango, there was one variety that was Ananta in Sirsi. So these attempt we made, and by the time I left, we had left around 10,000 hectares of total seedling and clonal orchards, which were sufficient to meet the demand of seeds and clonal propagation materials for nearly 50,000 hectares of our target. But suddenly, when I was Secretary Forest, the target went up to 1 lakh. We could not meet. Then we had to again buy seeds. Unless your materials are genetically proven, unless you know the source, unless you know what kind of material you are planting, I think it is not worth to use those materials in your plantation programs. So since it is a production, since our germplasms are very critical in not only conserving at the ex situ as well as in situ level, we also have to see that whether we can use the other means of trying to conserve and do cryopreservation of these kind of genes. So recently I was reading some articles, even herbarium collection, where I have nearly collected nearly 3,000 specimens of herbariums during my time. I was now we can really go back, pick up, we have got two sets of each herbarium. We can go pick up a piece of the leaf. We can do DNA analysis. So that is another way of doing your DNA preservation for the posterity. So therefore, the conservation strategy is very important. Now we have grown up production forestry, except in social forestry also in this thing. Particularly in Karnataka, I don't know about the other states. So we are now only focusing on the conservation aspect. And coming to animals, now we've got 9,000 species of animals in India. We've got 404 mammals. We have birds, which are very rich. And tiger conservation is one of the big success story in the entire world. The only place 90% of the tiger population in the entire world is only in India. And these out of 90, 25% are in Karnataka. We have nearly out of 50 tiger reserves, we got nearly 7 or 8 in Karnataka. And we have very good, once you eat the flagship species, once you conserve tiger as a flagship species, I am sure along with that, you conserve a lot of other mammals. lot of other Poaceae. See, Poaceae is another very important critical the genetic material, which is nearly I think 300 Poaceae species we have. Unless we conserve every Poaceae every grass, we cannot really protect or conserve our wild animals, because each species is has its own role in the protected areas. So we have nearly now 7 lakh hectares of protected areas in Karnataka. We are supposed to have I think 9 lakh hectares. But this the conservation of the mammals at the flag species level is one of the best the in situ conservation method of doing. Then coming to again back to flora, we MPCA, that is this medicinal plantation protected area, medicinal development area, is FRLST initiated the work. Then we did in 1995 extensive work, and nearly in I think 20 to 25 different agroclimatic regions, different forest types. We have established a very good MPC, MPDS. Every species has been tagged there. Every individual tree has been now named there, and we can always go back and see what's happening to those genetic processes. So genetic processes have to be I think we have to follow the mating system. What is the selection process that is going on? What is the competition that is going on? So what is how the characters are segregating, inheritance is happening. We have to estimate genetic advance data, genetic advance. Then you have to go for the or, this gene level studies of capturing what the DNA structure. I was trying to catch our participants' attention to two important things. That is, how do we really strategize our conservation program as committed under the Sustainable Development program by 2030, unless we try to go to the species level. Forest types are okay, we have gone. Now we should come to the species level. Then we have to go for this studies which are genetic processes which are inhibiting or promoting or coming in the way of regeneration. We have to study that. We do not have a capacity of taking up such studies of genetic process studies.